What's going on, guys? ErickMed here. In this video, we're going to be breaking down an MCAT section bank passage. Again, these section bank questions are of, a higher difficulty than the other, MCAT stuff like the question packs, the practice test, sample test, all that stuff. So these are a little harder than the ones before. So go ahead, guys, read through this passage, read through the questions, and attempt them on yourselves. Pause the video if you need to. So I'm going to scroll down here so you can see the whole passage. Okay, this is question 1, question 2, 3, 4, 5, and 6, and I believe that's it, right? Okay, yeah. I've never done this. I haven't done this like passage in so long. Last time I did this passage was like when I was studying for my MCAT, which was like probably like 9 months ago. So it's still pretty new to me, this passage here. Okay, so let's dive into it right now. All right, passage number 4. The majority of dietary vitamin A is stored in the liver and is then transported to several target organs as retinol by its carrier protein, retinol binding protein. Okay, this makes sense, retinol binding protein. In the liver, retinol forms a complex with RBP known as hollow RBP. STRA6 is the membrane receptor for RBP. Okay. STRA6 mediated retinol uptake is specific to RBP and uses a transport mechanism distinct from known transport mechanisms. Okay, so this is telling us that we're going to be learning something new here. Uses a transport mechanism distinct from known transport mechanisms. Okay, so we got to pay attention here. Hollow RBP binds to STRA6 transfected cells with high affinity. Only one retinol is provided to its receptor at a time, and RBP remains outside of the cell. Endocytosis inhibitors do not prevent STRA6 mediated retinol uptake. Okay, so this is what I'm going to highlight. I'm going to highlight this one. Okay, why am I highlighting this? Well, because sometimes these passages, they'll throw like a random fact at you. So here we're talking about, retinol, how it binds to the membrane, to hollow RBP, how this makes hollow RBP or the membrane receptor. Talking about the how this binding happens and how this uptake is going to happen. So, and then they just do this thing here called endocytosis inhibitors do not prevent STRA6 mediated retinol uptake. Like they just do this in here. It didn't really have to do much with the binding process because I feel like they're talking about so much binding here, and so they just do this random fact in here. And a lot of times when you're reading through these passages, guys, like you'll overlook something like this. And usually, 9 out of 10 times a question will have information that they just threw in this passage. See, look, let's see like with the next sentence. Upon uptake from hollow RBP, retinol is converted to retinol esters by lecithin retinol acetyltransferase or stored in the cell by cellular retinol binding protein 1. Okay, see? Again, they're going about how this retinol is bound and how it gets uptake into the cell. And this sentence as well talked about how it's being like inside the cell, how it's going inside the cell. And they just do this endocytosis inhibitors do not prevent this. So I'm going to highlight this because I might overlook this when we're going to a question. Researchers assayed the time-dependent uptake of radiolabeled retinol in untransfected cells, the control, and cells transfected with STRA6 alone, STRA6 and LRAT, and STRA6 and CRBP1. And then they give us a figure here. Okay, so for the figures, as I'm reading the passage, I don't look at the information inside the figure. Okay, so because a lot of times the questions that they give you, they can give you like a pseudo discrete question that has nothing to do with this figure. And you can, be wasting your time reading the figure, trying to understand it and try to interpret it, while a question could just ask you nothing about this. So only look at the information in the figure when the question asks for it. But you do look at the label of the figure just to get an idea of the experiment and what's going on in this passage. So time-dependent uptake of retinol in control and transfected cells. Okay, so here's the time, here's the retinol activity. Okay, that makes sense. So they're testing the uptake activity when, you have no STRE6 to control. You have STRE6 and these other proteins with them. Okay, makes sense. Not that bad. The researchers developed an assay to measure STRE6 mediated retinal release from hollow RBP. Okay, now we're testing the release. In this assay, different concentrations of hollow RBP were added at time 0 to membranes expressing STRE6 or STRA6/LRAT. Retinal fluorescence, which is enhanced when retinal is bound to RBP, was measured over time. Okay, retinal fluorescence in general, it's something that you have to be very aware of. You have to understand for the MCAT passages, for almost all of the MCAT. Okay, the whole biological and biochemical living systems section and the Chem and Phys section. They talk about fluorescence a lot. And what's the point of fluorescence? They usually use it in a bunch of experiments as like a tag or to measure and confirm your analysis of various research procedures and experiments and stuff. It's basically a marker. Okay, so their STRE6 and LRAT levels were the same in each reaction. Okay. So again, we have a figure here. I'm not going to waste time dissecting this figure yet. Retinal fluorescence assay in untransfected cells and cells transfected with STRE6 and STRE6/LRAT. Okay, I got a general sense of what this passage is about. I understand what this passage is about. So at the end of when you read a passage, try to get a little summary in your head. So my little summary in my head is that they're testing this membrane receptor. They're like, okay, can this membrane receptor, does it, uptake the activity of retinol binding protein, of retinol, into the cells? And is it more effective when this STRE6 membrane receptor is bound to LRAT, when it's bound to CRBP1? Is it more effective in releasing when it has different concentrations and it's with LRAT or not? Okay, so they're mainly talking about this receptor here. So let's go on to the question. Again, guys, the point of me dissecting these passages is to show you, look, this, got a grade change. Like this whole passage here, it wasn't that bad. Okay, it's not that hard. We broke this down and we know what this passage is trying to test for. We know what this experiment is about. So these questions should be relatively easy. Retinal uptake was measured in untransfected cells. Again, this is the control. And STRE6 transfected cells in the presence of retinol bound to either RBP or bovine serum albumin. Okay, they didn't tell me anything about BSA. Nothing in this passage was mentioned about BSA. So they want us, because they know we haven't seen this before. Okay, they know this person taking the test has never seen BSA before, but I want this person taking the test to make a decision about how BSA works. Uptake of retinol bound to RBP was also measured in cells that were co-transfected with a small inhibitor siRNA targeting STRE6. Which graphic shows the expected result of this experiment? Okay, small inhibitor. Boom, look what we highlighted. I told you this would come in handy. Okay, endocytosis inhibitors do not prevent STRA6 mediated retinal uptake. And we have a small inhibitor targeting STRA6. Okay, we have to make another decision here. Our first decision, these are two decisions this question wants us to make. Our first decision this question wants us to make is information about BSA. Is BSA good? Does it help? Is it bad? What is it? And the other decision this wants us to make is this siRNA, can this inhibit this whole STRA6 mediated retinal uptake? They tell us that endocytosis inhibitors do not prevent this uptake. So do we conclude that this siRNA is an endocytosis inhibitor and it does not prevent the uptake? Those are the decisions this question wants us to make. And if you're confident in your abilities, then you will make these decisions and you'll make them correctly. If you're not confident, then you'll probably, read a question and then try to convince yourself that this answer is correct when it's really not. So you have to be confident and you have to quote, put the axe down and say like, okay, this is wrong. I smell BS in this question in the answer choice, so I'm just going to eliminate it completely. I like this passage because it's a little more complicated. All right, but let's jump into it. We're already at like 10 minutes. Okay, so BSA, RBP, inhibitor. Okay, I'm making the decision that this inhibitor, this siRNA, is not an endocytosis inhibitor. It's not. It's an siRNA. And siRNAs are double-stranded RNAs that go ahead and they degrade other RNAs in a cell. So I'm going to conclude that this is not an endocytosis inhibitor. I feel like endocytosis inhibitor is more of a sort of like a blocking, sort of like it takes the place of something that wants to bind to it. But this siRNA is way different than an endocytosis inhibitor. So I'm going to classify this siRNA as an inhibitor that will decrease the retinal uptake activity. So which of these, correspond to my conclusion about this siRNA? So here we have question A, where it's negative, negative, positive. This is STRA6 siRNA. So STRA6 RNA is not added here. It's not added here, and it is added here. And from A, I can see that when we add this STRA6 siRNA, it doesn't make a difference here. When my conclusion is that it does inhibit it. So it would decrease the uptake activity. So A is not correct. Let's see. B, here's without, here's with. Okay, it does go down. So this could be correct. Again, here's without, here's with, it does go down. This can be correct. Again, without, with the siRNA, and it does go down. So B, C, or D. Okay, we have to make another decision here about this BSA. Bovine serum albumin. They didn't tell us anything about that. Nothing. How do you want us to make a decision about it? Well, we can compare RBP with bovine serum albumin. RBP or bovine serum albumin, are they the same at uptaking retinol, or are they different? Well, I think they're definitely not the same. I think they're different. I think RBP is way better at uptaking retinol than BSA. Why? Why am I making that conclusion? Well, because RBP literally stands for retinol binding protein. It has in the name retinol binding protein. Bovine serum albumin just doesn't. So retinol binding protein is very specific to retinol. So retinol binding protein should have a higher uptake activity than BSA. I don't know anything about BSA, but I do know that retinol binding protein is pretty good because it is specific to it. So I'm going to make this decision that RBP is way better than BSA. And B does not show that. B shows that BSA and RBP are the same for the uptake activity, which I don't agree with. So I'm going to go with C or D. Okay, what's the difference between C and D? Untransfected. All right, let's see here. Okay, this is pretty easy. If we have an STRA6 siRNA inhibitor, would that inhibit anything if we don't have STRA6? No. STRA6 siRNA inhibits STRA6. If a cell is not transfected with STRA6, then this STRA6 siRNA is not going to inhibit anything. There's no STRA6. Again, STRA6 siRNA, it inhibits STRA6. In the untransfected cells, in these black ones, there is no STRA6 receptor. So it's not going to inhibit anything. So I'm looking for something where there's no difference if we have STRA6 siRNA or if we don't have STRA6 RNA. If we, it doesn't make a difference because there's no target. Because they're untransfected. They don't have STRA6. So D. This should be correct. Let's see. Boom. Let's go. Okay, it's already 15 minutes. We gotta speed this up. Which statement is supported by the data shown in figure 1? Now we look at figure 1. Now we freaking dissect this whole thing. All right, I'm looking at this. I see that STRA6 LRAT is big. I see STRA6 RBP1 is smaller. This is smaller by itself, and the control is even smaller. STRA6 activity is associated with CRBP1 intracellular protein, but not the LRAT intracellular protein. It is associated with the LRAT protein. Look how big this is. You're telling me this is not associated? Like, no, get out of here. This is associated. This is associated. And you're telling me that it's not associated with LRAT? No, look how big this is. So this is wrong. STRA6 mediated retinal uptake in cells is only dependent on the ability to bind retinol. That's a very, again, if you see my last video, that's a very harsh conclusion. Only dependent on the ability to bind retinol? Yeah, that makes no sense. This is maybe a good answer. This is maybe like a okay answer, very okay answer, but it's not. What does this support? Nothing's supporting this. STRA6 mediated retinal uptake in cells is only dependent on the ability to bind retinol. I don't know. I don't know. Why? Why do I think this is bad? It's saying it's only dependent on the ability to bind retinol. It can bind, STRA6 can bind to LRAT, and we get a big, increase here. It can bind to CRBP1, and we get an increase from when it doesn't bind to STRA6 at all. So this is wrong. Neither LRAT nor CRBP1 are absolutely required for intracellular retinal uptake. This is a good answer. It's not like this really great answer, but it's a good answer. And it's way better than these two. Why? Because look, here's our control without STRA6. And here's our with STRA6. And we have some type of increase in retinal uptake activity. So we're uptaking retinol here. We don't necessarily need LRAT and CRBP1. They help a bunch. They help a lot, but we don't need them. STRA6 literally increases our uptake activity. So this is correct. It's not an amazing answer, but it is correct. Release of retinol from hollow RBP is most effective in the STRA6 only condition. Okay, that's literally about figure 2. We're talking about figure 1 here. So you're obviously wrong. Get out of here. And through process of elimination, C is probably the best answer. It's not an amazing answer, but it's way better than these two, and this one especially. So let's see. Boom, let's go. What is the dependent variable from the experiment shown in figure 2? Are you serious right now? Like, that is so, like, this is like a 5th grader question. What? What is the dependent variable from the experiment shown in figure 2? Okay, quick lesson here, guys. This is independent. This is dependent. Independent variable, dependent variable, dependent variable, relative retinal fluorescence. I don't even have to look at the other questions. I mean, the other answer choices. Like, what? Are you serious right now? Boom. That was such an easy question. See, guys, like, the MCAT is not that bad. It's really not that bad. This question was, oh my God, this question was like a 5th grader question. Which statement regarding STRA6 mediated retinal uptake is best supported by the passage? Okay. All right. Retinal uptake activity by STRA6 is limited without the presence of intracellular binding proteins. Wow. This sounds like a really good answer. STRA6 is limited without these proteins. Look, it's only here. We don't need these proteins, but we're limited. We can have these proteins and boom, look at that. But we are limited. This is a really good answer. Really, really good answer. Now, these other answer choices might be good answers, but this is a very, very good answer. The expression of STRA6 is likely to be higher in the liver than in target organs. Again, this could be correct. We don't know. There's literally no information supporting this. There's no information saying, oh, the other organs, the heart and the pancreas is very good at this. Like, no, what? They did not message this. It's not supported at all. This question wants what is best supported by the passage. This is not supportive at all. Get out of here. Retinal uptake activity by STRA6 is accelerated when retinol is stored as intracellular retinol than as retinal esters. Retinal esters, what the hell is that? Oh, okay. The LRAT LRAT makes retinol esters. Retinol esters increases uptake activity when it's a retinol ester. This is saying that when it's not a retinol ester, it's not accelerated. Well, LRAT makes retinol esters. When it's stored as retinol esters, look at this. It goes up. It's pretty high. So retinol esters is a good thing. It increases uptake activity. Intracellular retinol, it's okay. So we're accelerated when we are retinol esters. This question is saying we're accelerated, we're not retinol esters when we are intracellular retinol. So this is wrong, obviously. Binding of hollow RBP to STR6 is likely to have a Hill coefficient greater than 1. What? Alright, Hill coefficient, that's based on cooperativity. Greater than 1 means we have cooperativity. When it equals 1, that's like no cooperativity. And when it's less than 1, it's like negative cooperativity. What cooperativity is displaying here? This is, even if this was cooperativity, which is really not, there's no information supporting this. There's a lot of information supporting this. Look at this. They have this whole thing, this whole experiment. So A is the best answer here. Boom. Let's go. Okay, from the data shown in Figure 2, which statement best explains the relationship between STR6 and hollow RBP concentration in retinol release? Okay, they literally gave us a question here. Again, this is easy stuff. Why is this easy stuff? Because this question is literally like, okay, find this. From the data shown in Figure 2, so we're going to Figure 2, and Figure 2 only, which statement best explains the relationship between STRA6 and hollow RBP concentration in retinal release? Okay, the relationship between these two variables, STRA6 and hollow RBP concentration. That's all we got to find out. So let's see. STRA6 catalyzed retinal release increases as the hollow RBP to STRA6 molar ratio increases. Basically, we release more when our concentration of hollow RBP is higher. Is this true? Let's see. Here's STRA6 with the low hollow RBP concentration. And here's STRA6 with the high hollow RBP concentration. Okay, with the low RBP concentration, we release more than a high hollow RBP concentration. This graph here is a little tricky. It's a little tricky. Why? Because this is our control here, okay? We aren't releasing fluorescence. There's no fluorescence here. There is some fluorescence here, more fluorescence here, more fluorescence here, and the most fluorescence release here. Usually you're used to seeing, like, if a graph goes up, like here, that means, you know, we have more fluorescence release. You're usually used to seeing it like this. However, this is a little reversed because this is a relative retinal fluorescence. It releases, okay? So this is a little, I can see how it could be a little tricky here. You have to make that conclusion. So STRA6 catalyzed retinal release increases as the hollow RBP to STRA6 molar ratio increases. No. Lower concentration, we get a lot of release here. Higher concentration, we get very little release here. Retinal release in the STRA6 alone condition. Also, why is this more? It's because look at the time. Okay. In less time, we released all this stuff here than in more time when we release very little. Okay, retinal release in the STRA6 alone condition is not affected when the hollow RBP to STRA6 molar ratio changes. No, this is totally wrong. Why is this wrong? Look, high molar concentration, low molar concentration. You're telling me this is not affected? Look, this is very affected. Look how high this is and look how low this is. Get out of here. STRA6 becomes more dependent on LRAT for retinal release when the hollow RBP to STRA6 molar ratio increases. we're not talking about STRA6 becomes more dependent on LRAT for retinal release when the hollow RBP to molar content ratio increases. Okay, so let's look at LRAT. STRA6 becomes more dependent on LRAT for retinal release when the hollow RBP to okay. Here's STRA6 and LRAT with 4 micromolar of concentration. And here's STRA6 LRAT with 0.5 micromolar concentration. More dependent on LRAT for retinal release when the hollow RBP to STRA6 molar ratio increases. It was more dependent on LRAT for retinal release when the hollow RBP. This is saying when it increases, we're more dependent. No. Oh my god, sorry. Yeah, no. When it decreases the concentration, we're more dependent on it. If we're increasing the concentration when we have LRAT and STRA6, then we have less retinal fluorescence. We're not depending, we're more dependent when it decreases the concentration. And also, like, again, we're talking about STRA6 and hollow RBP concentration and retinal release. We're not talking about LRAT, so go away. Retinal release is only dependent on the presence of hollow RBP. Retinal release is only dependent on the presence of hollow RBP. That's wrong. This is wrong. It's only dependent on the presence of hollow RBP. No. Okay, this is a very interesting question here. Let me go back to this. Retinal release is only dependent on the presence of hollow RBP. This has to be wrong. This is a better answer than this, but this is wrong. This is wrong. This is literally wrong. It's only dependent on the presence of hollow RBP? No. Because we can have other stuff here that affects it. So this is wrong. This is an interesting answer choice, but I'm going to go with C. This is definitely wrong. These were definitely wrong. So through process of elimination, this is the better answer. So I'm going to go with C. Okay, that's right. You could look at this here, guys, if you want to understand more in detail. But for this one, we just literally do process of elimination here. Because I was going to decide between these two, but this one was so wrong that it had to be this one. Okay, the organ in which the hollow RBP complex forms also functions too. The organ is liver. This is a pseudo discrete question. You gotta know your content. Store bile? No. Secrete glucagon? No, that's the pancreas. Produce ACL? No, I believe that's the parietal or gastric cells of your stomach or whatever. Detoxify the drugs? Yeah, that's what the liver does. Okay, and that was it. You know, we got all of them correct. Let's go. But yeah, guys, just when you go through this passage, again, make sure you understand that this is a big dog with big bark but no bite. Okay, it can be a little complicated going through these, but it's really not. It's really not. Read slower. I recommend reading slower if you guys are, confused about the passage. Read a little slower and understand what the experiment is about. Understand what's going on. What are they, testing here? And also read the question carefully. Again, we read the question carefully here, and we had to make a decision. Make these decisions. Be confident in your decision, guys. They want you to make decisions in the MCAT. So we made our decision here. We made our decision here. Okay, that one was very easy. Again, this is easy stuff, guys. This is easy stuff. It's not that bad. It's not that hard. And this is the MCAT section bank, so it's supposed to be harder. And as you can see, as we dissected this down, it literally became like a piece of cake. So, yeah. So that's it for this video, guys. Let me know what you guys think. This is going to be the last one of these dissecting MCAT passages I'm going to be doing. Again, I don't know you do have to pay for these, so I don't know when they're gonna take it down. But like, share, subscribe, do all that fun stuff. Let me know what you guys think. Happy holidays. My Christmas was pretty lit. Oh yeah, it was pretty lit. I'm gonna say that it was lit. But yeah. So peace, guys.